So imagine stepping into this massive high-tech manufacturing facility,
But
but like there are no conveyor belts anywhere. There are no welding sparks flying around.
right, no heavy machinery.
Exactly, no massive mechanical presses stamping out sheets of metal. Actually, there's no metal in the room at all.
Which is weird for a factory.
Right. Instead, the center of the room is just dominated by this massive, completely clear vat, and it's filled with this dark, totally still liquid.
Just a giant pool of liquid.
Yes.
Yeah, I can picture it.
And here's the crazy part. Wherever the light actually touches the liquid, a solid, completely indestructible matrix instantly crystallizes.
Out of nowhere.
Literally out of the liquid. It is printing the reinforced hull of a spacecraft, layer by layer, out of the exact same proteins that cause mad cow disease.
I mean, when you describe it like that, it sounds completely hallucinatory.
Like, it sounds fake.
Yeah, like something pulled right out of a late-night science fiction paperback. But the thing is, the underlying physics, the biochemistry, the optical engineering required to actually build that room, it all exists right now.
And that is exactly why we are diving into this today. Because whether you're an engineer trying to, figure out the next paradigm of materials science, or maybe you're an investor looking at biotechnology.
Or you just want to know what the physical world is going to look like in 10 years.
Exactly. If you just have an insanely curious mind, this material is going to permanently alter your baseline assumptions. We are unpacking a really profound, wildly ambitious set of speculative science essays that were published in March 2026 by Zachary David Enfield.
Collectively titled From the Ground Up, the Unified Architecture.
Right. And I gotta say, reading through this, it is just a monumental piece of synthesis.
Oh, absolutely. Because Enfield isn't just pointing out like a few interesting lab experiments here and there. He is outlining a blueprint for a total systemic shift in human civilization.
Yeah, a complete teardown of how we do things.
Exactly. He's arguing that our current era, this mechanical extractive period where we mine stuff out of the earth, smelt it at thousands of degrees, stamp it, and then eventually just throw it in a landfill, he says that's a primitive dead end.
Right. He literally calls it a brute force approach to civilization. And so the core premise we're exploring in this deep dive is how we move from that brute force model to what he calls a fully programmable biological architecture.
It's a huge shift.
It really is. We are going to look at how the exact same logic used to cure genetic diseases at the microscopic level can actually be scaled up. Scaled up to manufacture those indestructible spacecraft hulls, or build supercomputers out of DNA.
Or even fundamentally rebuild our collapsing agricultural ecosystems.
Right. So, okay, let's unpack this, because the scope of Enfield's architecture is just absolutely massive.
It really spans from individual atoms all the way up to planetary scale terraforming.
Yeah, and before we get into all that, what's the guiding principle here? Like, what does the listener really need to keep in mind as we go through this?
The main thing I want you to hold on to as we navigate this, because, it applies to everything from agricultural runoff to those light-activated proteins, is that we have to fundamentally change our definition of biology.
Okay, how so?
Well, for centuries, we've basically treated biology either as a static resource to be harvested, like logging a forest,
Right.
or as this wild, untamed enemy that we have to suppress, like a virus.
Yeah.
But Enfield demands that we start treating biology as an information processing technology.
Like a computer.
Exactly. A cell isn't just a squishy bag of chemicals. It is literally the most sophisticated, energy-efficient computational substrate in the known universe.
Because it runs code.
It runs code. And once you truly understand the code, you can rewrite the physical world.
Okay, I love that. So let's start at the absolute largest scale to really grasp how these systems connect. Let's look at the planetary landscape.
The macro scale.
Yeah. Early in the essays, Enfield introduces this concept he calls the linear fallacy. And when I was reading this section, it just struck me as such a devastating critique of our entire modern economy.
Devastating is definitely the right word for it. The linear fallacy is basically this arrogant assumption that we can take the incredibly complex, closed-loop cycles of the Earth and treat them as simple, linear transactions.
It's like a straight line.
Right. In a linear system, you start at point A, extract some raw material, move it to point B to process and consume it, and then whatever's left over, you discard at point C as waste.
You're just constantly drawing down from a finite source and filling up a finite sink.
Exactly. It is a one-way street toward total entropy. But nature, on the other hand, absolutely abhors a one-way street. Every single biological system that has survived deep time operates in a closed loop.
Where the waste from one process is just the fuel for another.
Right. The output is the essential metabolic input for the next thing.
And the data he uses to illustrate the consequences of our linear thinking is just terrifying. He points to the Ogala Aquifer.
Oh yeah, that section is bleak.
For those who aren't familiar with it, the Ogala is this massive, shallow water table sitting right beneath the Great Plains of the United States. It stretches across, I think, 8 different states.
Yeah, it's huge. But the geological history is the crucial part.
Right. This water didn't just rain down last spring. It took anywhere from 6 to 12 million years to fill up.
Millions of years.
Mostly from the melting of glaciers retreating after the last ice age. So it is literal fossil water.
Which means it is, for all practical purposes, a completely non-renewable resource on any kind of human timescale.
Exactly. And yet today, the Ogala provides roughly 30% of all the groundwater used for irrigation in the entire U.S.
We're just pumping it out.
We are pumping it out with these massive center pivot irrigation systems to grow corn, wheat, soy. And because we treat it as this infinite linear resource, Enfield points out a really scary stat.
The Texas Panhandle data.
Yes. In specific, heavily farmed regions like the Texas Panhandle, up to 70% of the aquifer could be functionally unusable in just 20 years.
It's staggering.
We are going to completely drain what took millions of years to accumulate in the span of just one single human lifetime.
And we're doing all this to support an agricultural model that relies incredibly heavily on synthetic fertilizers, which is just another perfect catastrophic example of this linear fallacy.
Oh, the fertilizer mass balance. Walk us through that.
Yeah, look at the mass balance of our agriculture. Every single year, the U.S. applies 12 million tons of synthetic nitrogen and 4 million tons of phosphorus to its crops.
That's a lot of fertilizer. Where does it even come from?
Well, the nitrogen is largely pulled right out of the atmosphere using the Haber-Bosch process. That requires extreme heat and pressure, so it consumes just massive amounts of fossil fuels. And the phosphorus, that's literally just mined directly out of the ground.
So we spend all this energy, all this capital just to create this stuff, and then we just spray it all over the fields.
Right. But plants aren't perfectly efficient machines. They can't possibly absorb all of it.
No, of course not. So a massive percentage of that nitrogen and phosphorus just sits there in the soil. And then the next heavy rain comes along, and it washes directly into the drainage ditches,
Which flow into the streams.
which empty into the rivers, specifically the Mississippi River Basin.
Right, and all of that incredibly nutrient-rich runoff from basically the entire American Midwest funnels down the Mississippi and just empties straight into the Gulf of Mexico.
And when those nutrients hit the warm ocean water, they do exactly what fertilizer is designed to do, right?
They stimulate massive plant growth.
But in the ocean, it's not corn, it's phytoplankton and algae. So you get these explosive algal blooms that just blanket the surface of the water.
And when those billions of algae inevitably die, they sink to the bottom. And then bacteria start decomposing them. But that decomposition process consumes oxygen.
It literally vacuums the dissolved oxygen right out of the seawater.
Exactly. The result is total hypoxia. Enfield cites the staggering size of this. It's a 6,000 square mile dead zone in the Gulf of Mexico where marine life literally just suffocates.
It's mind-boggling. I mean, we are basically treating our ecological bank account like an infinite credit card. We're making these massive multi-million-year withdrawals from aquifers, and our only deposit is a toxic sludge that kills the ocean.
It is the ultimate failure of mass balance accounting.
Okay.
We are liquidating our natural capital and just writing it down on the balance sheet as agricultural income.
Which is a terrible business model.
But this is exactly where Enfield pivots from despair to his proposed architecture. Because if the fundamental problem is that we have severed the biological loops, the solution isn't to just, use slightly less fertilizer.
Or build a more efficient water pump.
Right. The solution is to deliberately rebuild the closed loops using biological infrastructure.
Oh.
And that brings us to two deeply interconnected macrosystems he outlines: the duckweed loop and bio-CCS.
Bio-CCS being bioenergy with carbon capture and storage using algae. Okay, I have to admit, when I saw that a major pillar of human civilization's future rested on duckweed, I actually laughed out loud.
I think a lot of people probably do.
Right. Because I've always just thought of duckweed as that annoying green pond scum that ruins decorative fountains.
It definitely has a PR problem.
It really does. But the biology of this plant is actually miraculous. Duckweed, or Limnaceae, is literally the fastest growing vascular plant on Earth.
Because it's so simple.
Yeah, it doesn't waste any energy growing deep roots or thick, woody stems. It's essentially just a tiny, floating leaf. And because of that simplicity, under optimal conditions, duckweed can double its entire biomass in just 16 to 48 hours.
Just think about the math of that exponential growth for a second. Let's say you have a single square meter of duckweed on Monday.
Okay.
If it doubles every 2 days, you have a massive, dense carpet of biomass covering a huge area in a matter of weeks.
It's explosive.
But the crucial factor isn't just how fast it grows. It's what it actually uses to fuel that growth. Duckweed evolved to thrive in highly eutrophic water.
Eutrophic meaning water that is completely overloaded with nutrients.
Right. Its primary food sources are nitrogen and phosphorus.
Wait, the exact same elements destroying the Gulf of Mexico?
Precisely. So Enfield proposes integrating duckweed directly into the agricultural wastewater stream.
Oh, I see.
If you channel agricultural runoff or even municipal wastewater through shallow, slow-moving duckweed ponds, the plant acts as a hyper-efficient biological filter.
Wow.
Studies show it can strip up to 96% of the phosphorus and massively reduce the nitrogen load from the water before it ever even reaches a river.
But it doesn't just make the pollution disappear magically, right?
No, it metabolizes it. It converts that toxic runoff into dense, harvestable plant tissue.
And then Enfield pairs this with those algae bioreactors we mentioned. He outlines this system where you take the actual raw flue gas from a coal power plant or an ethanol refinery, and you pipe it directly through a water column filled with microalgae.
Because microalgae are single-celled organisms, so they are just the ultimate photosynthetic engines. They don't have roots or stems either.
Right, they just exist to process light and carbon.
Yeah, they absorb CO2 directly from that industrial exhaust, use sunlight, and convert that carbon almost entirely into lipids, into oils.
And the efficiency gap between this and traditional agriculture is just staggering. If you plant an acre of soybeans, you might extract, 48 gallons of usable oil in a year?
Yeah, somewhere around there.
But an acre of optimized, densely cultivated algae can theoretically yield 10,000 gallons of oil per year.
It's not even close.
And all while pulling 12 tons of CO2 out of the atmosphere, you are taking literal industrial pollution and biologically transmuting it into a high-yield biofuel feedstock.
So if you put these pieces together, Enfield is proposing we completely redesign the physical layout of our agricultural landscape. He introduces this concept called Ditch 2.0, or hybrid irrigation.
Okay, what is Ditch 2.0?
Well, for the last 50 years, we've been replacing old, leaky, gravity-fed irrigation ditches with highly efficient pressurized pipes.
To save water.
Right. But those old, leaky ditches actually served a really vital ecological purpose. They allowed water to slowly seep back down and recharge the aquifers.
Oh, interesting.
So Enfield says we need to intentionally rebuild a network of shallow channels alongside our fields, but engineer them specifically for duckweed cultivation.
So these channels catch the fertilizer runoff.
The duckweed rapidly absorbs the nitrogen.
And then you harvest the duckweed until it right back into the soil as a slow-release biofertilizer.
Exactly. You have taken a toxic waste stream and turned it into a closed, profitable loop. It completely upends how we define waste.
Yeah, because if you have a ton of nitrogen flowing down the Mississippi River, it is a poison. It's a massive liability.
But if you have that exact same ton of nitrogen captured inside the cellular walls of a duckweed harvest, it is free fertilizer.
That is such a good way to look at it. The atoms are completely identical. The only difference is whether those atoms are participating in a broken linear system or a closed biological loop.
Precisely.
Okay, but I was reading this section on the nutritional profile of duckweed, and it's pretty incredible. Depending on the strain, duckweed can be 33 to 44% crude protein by dry weight.
It's incredibly nutrient dense.
Right, so it is this rich, nutritious feed for livestock or poultry or farmed fish. But here's the really obvious problem I saw immediately.
The contamination risk.
Yes. If we are growing this hyperabsorbent plant in feedlot runoff or literal municipal sewage just to clean the water, you absolutely cannot feed sewage-grown plants to a dairy cow.
No, absolutely not.
The pathogen risk alone, not to mention heavy metal accumulation, it would just be a public health nightmare.
It would be, and Enfield doesn't gloss over that at all. He addresses it with what he calls the two-stream principle. The whole architecture relies on absolute, rigorous segregation of the inputs.
Okay, break down the two streams.
Stream A is the sanitation loop. This is the duckweed grown in municipal sewage or heavy industrial runoff or those concentrated animal manure lagoons.
The nasty stuff.
Right. The biomass harvested from Stream A is never, ever allowed into the food chain. It is used exclusively for soil amendment, composting, or it is fed into anaerobic digesters.
Where bacteria basically break it down into methane biogas for electricity.
Exactly. And the intense heat and the biological activity of those specific processes completely destroy any human pathogens.
Okay, so Stream A handles the hazardous waste and basically turns it into power and soil. What about Stream B?
Meanwhile, you operate a completely distinct Stream B. This is the duckweed grown in clean, controlled agricultural runoff or specially designed clean water raceways.
Got it.
Because the input water is safe, the harvested biomass is safe. So Stream B is what provides that incredibly dense 44% protein feed for livestock and aquaculture.
So the logistics have to be completely bulletproof, but the biology itself is doing all the heavy lifting. Zero cross-contamination. It makes so much logical sense. I mean, you look at these systems using duckweed to close the fertilizer loop, using algae to eat industrial carbon and spit out thousands of gallons of biofuel, and you really have to ask,
Why aren't we doing this everywhere?
yes, if the biochemistry is this clear and the economics are this favorable, why aren't there duckweed channels and algae scrubbers outside literally every farm and factory in the country right now?
Because the barrier isn't scientific. It is a massive institutional failure.
The bureaucracy.
Yeah, our entire modern infrastructure is built in highly specialized, completely isolated silos. The sanitation department's only metric for success is managing wastewater compliance.
They just want to hit their EPA target.
Right. And the Agricultural Extension Office, they only care about crop yields and synthetic fertilizer application. The Department of Energy manages the coal plants.
So they don't even talk to each other.
These bureaucracies literally do not speak the same language. If you go to a conventional wastewater plant manager and propose replacing their expensive, predictable chemical flocculants with, like, a massive, living, seasonal pond of floating plants, they will look at you like you are insane.
Because they want chemical certainty.
Exactly. A biological system, even if it is a fraction of the cost and produces a super valuable byproduct, introduces variability. Implementing the unified architecture requires a total paradigm shift. We have to stop thinking like civil engineers pouring concrete and start thinking like systems ecologists.
That makes total sense. And to really think like an ecologist, to actually build these macro-level planetary loops, Enfield argues that we have to deeply understand and manipulate the underlying operating system that governs that algae and that duckweed.
Right, you can't just harvest nature anymore. We have to author it.
Which means we have to zoom all the way in. We have to go from the scale of a landscape all the way down to the scale of a single cell.
Because the photosynthetic efficiency of that algae, or the exact rate at which that duckweed absorbs phosphorus, is dictated entirely by its DNA.
And this takes us to the second major pillar of the architecture, debugging the human machine.
Yes, the microscale.
I love that specific phrasing, debugging. Enfield points out how deeply entrenched our linguistic metaphors are when it comes to biology, particularly with human health. We almost exclusively use the language of warfare.
We fight cancer.
We battle infections.
Yeah.
Viruses invade our borders. We essentially view our bodies as a battlefield.
It is an incredibly pervasive framing, and he argues it fundamentally limits how we approach medicine. Think about it. If you are fighting a war, your primary tools are weapons.
Right.
You use a scalpel to physically cut the enemy out, or you use chemotherapy to just carpet bomb the terrain and hope the cancer dies before the patient does.
It's brute force again.
Exactly. But if you accept Enfield's premise that a living cell is the most sophisticated computational substrate ever to evolve, then a disease isn't an invading monster. Disease is just corrupted code. Cancer is essentially just a growth regulation loop that somehow forgot its stop command. A genetic disorder is just a spelling error in a string of syntax.
So if the problem is corrupted information, the most elegant solution isn't to bomb the hard drive.
The solution is a software patch. You just need to debug the code.
But obviously you can't just plug a USB cable into a human liver to upload a patch. You need a delivery mechanism capable of navigating the incredibly hostile environment of the human bloodstream, finding a specific cell, bypassing its security membrane, and depositing a payload of new code directly into the nucleus.
Which sounds impossible.
Right, but it turns out nature already spent 3 billion years perfectly engineering the ultimate molecular USB drive.
the virus.
The virus. This requires such a massive cognitive shift for most people. We're so conditioned to view viruses as the ultimate biological enemy.
Especially recently.
Yeah. But stripped down to its purely mechanical essence, a virus is literally just a courier. It is an extraordinarily elegant protein shell designed to protect a payload of genetic instructions.
And over billions of years of ruthless evolutionary pressure, viruses have basically solved the hardest problems in nanoscale logistics.
Right.
They know exactly how to evade immune detection
They know how to dock with highly specific receptors on a cell membrane.
and how to trigger the cell to engulf them. They are literal delivery drones.
The only reason a virus like, say, influenza or HIV makes us sick is because of the payload it's carrying. Its code says, hijack this cell's machinery to print 1 million more copies of me and then destroy the cell.
But scientists realized, what if we just pop the hood?
Yeah. What if we hollow out the virus, rip out the destructive replication code, and replace it with a constructive healing payload?
Enfield calls these engineered couriers viral nanites.
And I want to emphasize, this is not speculative future casting. This is happening right now in clinical settings.
Yes, the most common chassis for these nanites right now is the adeno-associated virus, or AAV. It is a small, completely harmless virus that doesn't cause any disease in humans, but it is incredibly efficient at infecting cells.
So we strip out its viral DNA and load it with an editing mechanism, most notably CRISPR-Cas9.
Let's actually walk through how CRISPR works, because when you look at the mechanics, it perfectly fits this software patch analogy.
Okay, break it down.
CRISPR isn't just some chemical drug that floods your system. It is a highly precise molecular text editor. It basically consists of two parts. First, you have a guide RNA. Think of that like the Ctrl+F search function on your keyboard.
Okay, so it searches the DNA.
Right. It scans the billions of letters of DNA in the cell until it finds the exact specific sequence of corrupted code. And once it finds it, the second part, the Cas9 enzyme, comes in. That acts like a pair of molecular scissors.
It makes a precise cut.
Yes, snipping out the bad code
Hmm?
and allowing the cell to repair the sequence with the correct information.
The precision of that is just astonishing. We are talking about finding and replacing a single misspelled chemical letter out of 3 billion.
And the clinical data Enfield provides here is staggering. For example, in December 2023, the FDA approved a therapy called Casgevy. It is a treatment for sickle cell disease.
And sickle cell is a brutal disease.
Brutal, agonizing. And it's caused by a single tiny mutation in the DNA that causes red blood cells to fold into a stiff sickle shape that causes massive blockages and pain. For decades, all we could do was manage the pain.
But Casgevy uses CRISPR to target and edit a specific gene called BCL11A, right?
Exactly. By tweaking that one line of code, it essentially tells the body to start producing a different, healthier form of fetal hemoglobin.
And the results were amazing. In the clinical trials, 29 out of 31 patients who received this treatment were completely free of severe pain crises for at least a year.
They didn't take a pill every day to manage symptoms. They received a permanent software patch right at the source.
And the velocity of this technology is accelerating so rapidly because, like, Casgevy required an ex vivo approach, right? Meaning they had to physically extract the patient's bone marrow stem cells, edit them in a lab, and then transplant them back in.
Which is incredibly expensive and complex.
Yeah. But Enfield points to a clinical trial that launched at the Cleveland Clinic in November 2025 for a therapy called CTX310. This is moving to an in vivo approach.
Yes, this is a huge leap. They use lipid nanoparticles, the exact same microscopic fat bubbles used to deliver the mRNA COVID vaccines, to carry the CRISPR editor directly into the patient's bloodstream.
So no marrow extraction.
None. The lipid nanoparticle specifically targets the liver, enters the cells, and permanently switches off a gene called AGPTL3.
And the result of just turning off that one single gene?
A 50% reduction in LDL cholesterol from a single intravenous infusion.
Wow. Just imagine replacing a lifetime of taking daily statin pills with all their side effects with a one-time genetic update that permanently optimizes your liver's cholesterol processing.
We are moving from external management to internal compilation.
Okay, but here's where it gets really interesting and profoundly weird, where Enfield really pushes the boundaries. Because if we have the delivery drones, the lipid nanoparticles and viral nanites, and we have the text editor CRISPR, why restrict ourselves to just fixing broken human code?
This is the leap.
Right. Evolution has been running a 3 billion-year planetary scale beta test. Every species on Earth contains genetic subroutines that solve complex physical problems. Enfield calls this functional translation.
An amazing concept.
If we can read the code of other species, can we translate it, compile it, and run it on human hardware?
This is the critical threshold where we cross from therapeutic healing to intentional biological augmentation.
Yes.
Nature is this vast open-source library of extreme survival code. Consider the tardigrade, the microscopic water bear. These organisms are famous for surviving conditions that would instantly liquefy human DNA.
They can survive the vacuum of space, extreme dehydration.
And radiation doses thousands of times higher than the human lethal limit.
See, when I first read about this, I just assumed they had incredibly thick outer shells or something. But the mechanism is actually entirely internal, right?
Yes. Researchers discovered that tardigrades produce this unique protein called DSIP, which stands for damage suppressor.
Okay, what does it do?
It relies on electrostatic interactions to physically bind to the tardigrade's DNA, essentially wrapping it in a protective molecular shield. This shield prevents radiation-induced double-strand breaks, but somehow without interfering with normal cellular functions.
It is an incredibly elegant piece of evolutionary engineering.
And scientists didn't just marvel at it, they actually sequenced the code for that DSUP protein. And Field outlines how we could use functional translation right here.
So you take the genetic blueprint for the tardigrade's radiation shield,
You translate it into a human-compatible mRNA sequence.
package it in a lipid nanoparticle, and deliver it to human cells.
Exactly. Suddenly, those human cells begin manufacturing the tardigrade protein, wrapping their own DNA in the shield.
Just imagine the applications for that. You could give this transient mRNA patch to a cancer patient right before they undergo intense, targeted radiotherapy, completely protecting their healthy tissue from collateral damage.
Or you could give it to astronauts facing intense cosmic radiation on a multi-year mission to Mars.
And because it's mRNA, you aren't permanently changing their genome. You are just temporarily downloading an app from a water bear to give a human temporary radiation resistance.
It fundamentally changes our relationship with the biological world. We are no longer limited by our own slow evolutionary timeline. Or, consider a more structural example he gives, the axolotl, a species of salamander.
Oh, the regeneration stuff.
Yes, if an axolotl loses a leg, or even part of its heart or spinal cord, it doesn't form a scar. Instead, the cells at the site of the injury actually de-differentiate.
Meaning what, exactly?
They revert back into stem cells, forming a mass called a blastema, and then they completely regrow the bone, muscle, nerve, and skin perfectly.
And Enfield's argument here is that humans didn't necessarily lose this ability. We share a massive amount of our genetic lineage with these amphibians.
Right, the genetic toolkit, the blueprint for building a limb, is obviously inside us. I mean, we built our limbs when we were embryos.
We just turned the program off once we were fully grown.
Exactly. Evolution prioritized fast scarring to prevent infection over slow regeneration. But Enfield suggests that the code for regeneration is still sitting dormant in our hard drives. We just need the right viral nanite to go in and flip the regulatory switches back on.
Okay, wait, I have to stop you here because the alarm bells in my head are absolutely deafening right now.
That's a normal reaction.
We are talking about injecting engineered viruses and nanoparticles into healthy people to fundamentally rewrite human DNA. We are talking about deliberately turning off cholesterol-regulating genes and intentionally reactivating dormant explosive cell growth programs to regrow limbs.
It sounds incredibly risky.
If you tell a localized group of cells to rapidly divide and grow, and that code goes rogue, or it forgets to turn off, that is literally the exact mechanical definition of cancer, unchecked cellular proliferation. How in the world can this possibly be safe?
That is the absolute existential engineering question of synthetic biology. And Enfield addresses it head-on in the text. You cannot introduce programmable logic into a living system without a hardwired abort sequence. The architecture demands the mandatory inclusion of kill switches at the genetic level.
But how exactly do you build a biological kill switch? It's not like you can install a physical button.
Use synthetic promoter sequences and allosteric regulation.
Okay, unpack that for us.
A promoter is a region of DNA that initiates the transcription of a particular gene. You can engineer the viral payload so that the editing program is completely inert by default. It only activates under highly specific, tightly controlled conditions. For instance, you can program the nanite to automatically self-destruct after exactly 3 cell divisions, preventing runaway growth.
Oh, so it has a built-in biological clock.
Exactly. Or, for even more control, you use a small molecule switch.
How does that work?
The genetic edit is successfully integrated into the patient's cells. But the new protein, say, the limb regeneration sequence, remains folded up in an inactive state. It can only become active if it physically binds to a specific synthetic chemical compound.
Like a key.
Exactly like a key. The patient has to physically swallow a specific pill that contains this chemical key. The chemical enters the bloodstream, binds to the engineered protein, changes its shape, and turns the growth program on.
And the second they stop taking the pill?
The chemical washes out of their system, the protein reverts to its locked state, and the cell growth program instantly shuts down. The reversibility and the fail-safes have to be built into the very architecture of the molecule.
That is fascinating. Okay, so we have the code DNA, which operates on quaternary logic and is basically the densest information storage medium in the universe.
Right.
And we have the couriers, the viral nanites and lipid nanoparticles perfected over billions of years to deliver that code precisely. But up to this point, we have only been talking about squishy internal biology, fixing a liver, regenerating tissue.
The soft stuff, right.
How do we take this paradigm of programmable biology and use it to build the external world? If I want to build a bridge or a microchip or the hull of a spacecraft, I can't build it out of soft liver tissue. We need a universal, rigid, structural building material.
Which brings us perfectly to the third major synthesis in Enfield's work, the material scale. If biology is actually going to replace mechanical industry, it needs industrial-grade material.
And to find it, Enfield turns to what is arguably the most terrifying apocalyptic biological phenomenon known to modern medicine.
Prions.
Yeah. When I hear the word prion, my mind instantly goes to mad cow disease or Creutzfeldt-Jakob disease. It is the ultimate biological horror story.
It really is.
It's a disease that literally turns your brain tissue into a sponge, causing rapid dementia and death. It's 100% fatal, and it's practically invincible.
And it is deeply terrifying in a medical context precisely because of its structural resilience. To understand why Enfield is so fascinated by them, we have to look at what a prion actually is.
Oh, yeah.
It is not a virus. It is not a bacteria. It contains absolutely no DNA or RNA. A prion is just a protein. Proteins are long chains of amino acids that fold into specific three-dimensional shapes to do their jobs. But a prion is a protein that is folded incorrectly.
And the terrifying part is that this misfolded shape is somehow contagious.
Exactly. The prion acts as a physical template. When this misfolded protein bumps into a healthy, normally folded version of the same protein, it physically interacts with it, forcing the healthy protein to unravel and snap into the misfolded shape.
So 1 becomes 2, 2 become 4, 4 become 8.
It is a runaway, cascading structural corruption. And because they lack genetic material, you cannot kill them with radiation or UV light. Because they are so tightly folded, they survive boiling temperatures and harsh chemical disinfectants that would obliterate normal viruses.
But Enfield looks at this apocalyptic, brain-destroying protein and says, wait a minute, look past the disease. Look at the mechanics.
He strips away the pathology.
Right. He points out that the underlying physical structure of a prion, what biochemists call the cross-beta amyloid scaffold, is actually an absolute engineering marvel. If you engineer a synthetic version of this, stripped of its ability to cause disease in humans, you have a self-assembling, self-replicating, indestructible nanocomposite.
And what's fascinating here is understanding why that cross-beta structure is so incredibly robust.
Yeah, what makes it so tough?
In a normal protein, you have various shapes like loose loops or alpha helices. But in an amyloid prion, the amino acid chains lay out flat against each other in rigid sheets called beta sheets. These sheets are bound tightly together by a massive network of hydrogen bonds along the backbone. And sticking out from these sheets are the side chains of the amino acids, which essentially interlock like the teeth of a zipper, acting as molecular shock absorbers.
So it creates a structure that is both intensely rigid, yet capable of dissipating mechanical energy perfectly.
Exactly. Enfield talks about this metric called hydrogen bond density, or HBD. By precisely tweaking the amino acid sequence to maximize that HBD, materials engineers can tune this amyloid material across a breathtaking spectrum of physical properties.
I was looking at the stats Enfield provided on this, and they are wild. By optimizing that cross-beta structure, these synthetic prion fibers reach a toughness, meaning their ability to absorb energy and deform without fracturing, of 160 megajoules per cubic meter.
Which is astronomical.
To put that in perspective for you, that significantly beats the toughness of spider silk, which is already considered nature's miracle material.
And it achieves that extreme toughness at roughly 1/8 the physical density of steel.
It is ultra strong, ultra lightweight, and instead of requiring a massive carbon-spewing smelting plant to produce, it literally grows itself exponentially in a vat of water at room temperature.
And because of that dense hydrogen bonding network, it is practically impervious to environmental degradation. You can boil these structures in pure formic acid. You can bathe them in harsh enzymatic detergents.
You can blast them with levels of gamma radiation that would instantly fry the solid-state logic of a silicon computer chip.
And the amyloid scaffold remains completely intact. But its structural resilience is really only half the story here. Enfield is equally focused on its astonishing electrical properties.
Oh man, this was the part that truly blew my mind. It's like finding out that the creeping rust slowly destroying your car is actually the strongest, most highly conductive building material in the universe, provided you know how to sequence it correctly.
Right, because these proteins don't just sit there passively, they conduct electricity.
They do, and the quantum mechanics behind it are just beautiful.
Within the core of that tightly packed amyloid fiber, you have specific amino acids like tyrosine and phenylalanine. These are aromatic amino acids, meaning they have ring-like structures containing delocalized electrons.
Okay, science term, but basically the electrons can move around easily.
Yes. Because the beta sheets pack these rings so incredibly close together, their electron orbitals physically overlap. This creates a continuous, highly conductive pathway right down the center of the fiber. It allows electrons to travel down the protein via quantum tunneling.
A process called tyrosine hopping, right?
Exactly.
So you essentially have a microscopic, self-assembling biological wire that intrinsically conducts electricity without needing any coating of copper or gold.
And beyond just passive conductivity, these structures exhibit strong piezoelectricity.
Piezoelectricity, meaning they can convert mechanical stress into electrical voltage and vice versa, kind of like the crystal in a quartz watch.
Precisely. Researchers have actually engineered specific amyloid beta nanotubes that exhibit incredibly high piezoelectric coefficients.
Okay, what does that mean in practice?
It means you can expose these microscopic protein tubes to ambient ultrasound waves, just sound waves passing through the liquid. And the physical mechanical squeezing of the sound wave causes the amyloid to generate up to 1 volt of electricity locally.
Just from sound.
Just from sound.
Yeah.
Think about the medical applications of that. You can inject these engineered non-toxic amyloid fibers directly into a tumor. Then, from the outside of the body, you just hit the patient with a standard ultrasound wand.
Oh, wow.
The sound waves cause the fibers inside the tumor to generate a localized voltage spike. That voltage reacts with the surrounding water to create reactive oxygen species, essentially highly corrosive free radicals, which rip the tumor cells apart from the inside out.
That is incredible. Wireless, targeted cancer destruction driven entirely by self-assembling proteins and sound waves.
It is. But Enfield pushes this even further into what has to be the most mind-bending concept in the entire collection of essays, DNA prion hybrid computing.
We are talking about using these exact materials to completely supersede the silicon microchip.
This is where we leave the limitations of traditional digital architecture completely behind. Our current computers are binary, right? They use transistors that act as simple switches, either on or off, a 1 or 0. It's base 2 logic.
Right.
But DNA is intrinsically a quaternary system. It utilizes 4 distinct chemical bases: adenine, cytosine, thymine, and guanine. It's base 4 logic.
So mathematically, at equivalent clock frequencies, a quaternary system can transmit exponentially more information per physical operation than a binary system.
And the physical storage density is almost incomprehensible. A single gram of DNA can theoretically store hundreds of exabytes of data.
He notes that all the digital data currently existing in the entire world could fit in a space the size of a shoebox if it was stored in DNA.
Exactly. But historically, the massive roadblock to DNA computing has always been speed. It relied on incredibly slow fluidic chemical reactions to read and write the data. It was dense, but it was so sluggish.
Right, until we introduced the catalyst Enfield highlights, terahertz radiation.
DNA molecules are not static things. They are dynamic, vibrating structures. And it turns out that the hydrogen bonds holding the 2 strands of the DNA double helix together have natural resonant frequencies right in the terahertz range.
So by hitting a DNA molecule with highly precise, intense pulses of terahertz radiation, scientists can essentially ring the DNA like a bell.
They can resonantly unzip the 2 strands apart and then re-zip them together. And they can do this in picoseconds. trillionths of a second.
So the terahertz radiation acts as the clock speed. You now have a quaternary memory node, the DNA that can be read, written to, and altered at blistering speeds.
But how do you connect them?
Right, you can't exactly wire millions of microscopic DNA molecules together with copper wire. Copper is way too massive at that scale. You can't use silicon traces.
You use the prions, the conductive amyloid fibers.
Boom.
They are roughly 10 nanometers wide. You genetically program the amyloid monomers to specifically bind to the ends of the DNA nodes. When you trigger the self-assembly cascade, the indestructible electrically conductive prion fibers grow outward, physically bridging the gaps between the DNA molecule.
They become the nanoscale wiring.
What you end up with isn't a rigid, grid-like silicon chip. It is a computational soup. A three-dimensional neural network where DNA stores the memory and performs the quaternary logic. The amyloid fibers act as the conductive synapses connecting them, and the terahertz radiation drives the clock speed of the entire liquid computer.
Okay, my brain is officially melting a little bit here. We are talking about building hyper-dense molecular supercomputers out of disease proteins and genetic code just floating in a vat.
Why?
But as I read this, I kept hitting a massive mechanical wall. I had a major pushback here regarding how we actually utilize this stuff.
Okay, what's the issue?
You described the prion cascade earlier. You drop a seed into a vat of liquid monomers, and it forces them to misfold, growing exponentially in all directions.
Right.
If it just grows outward randomly like a crystal, how do you actually shape it? A vat filled with ultra-strong, highly conductive protein goop is scientifically amazing, but it is entirely useless as a structural object. You can't build the intricate architecture of a microchip out of a random blob.
No, you can't.
You can't build a car chassis out of goop. How do you dictate the geometry?
It is the perfect question, and it identifies the exact historical limitation of this entire field. It's a fundamental problem of control. You have the ultimate bioink, but you have no pen.
Right. How do you steer the self-assembly?
You need a mechanism to tell the protein exactly where to form the rigid structure and exactly where to remain a liquid. And this leads us to section 4 of the architecture, the fabrication scale. To exert spatial control over biology, Enfield introduces a technology called prion optogenetic lithography.
Optogenetic lithography. So to turn this biological soup into precise engineered 3D structures, the steering wheel is light.
Exactly. Light. Enfield is synthesizing 3 distinct scientific disciplines here to create a completely new manufacturing paradigm.
Okay, what are the 3?
First, you had the prion cascade we just covered that provides the indestructible material and the self-amplifying growth. Second, you integrate optogenetics.
Optogenetics meaning?
This involves light-responsive proteins found in nature, like Cry2 or LOV2. These are proteins found in certain algae or plants that physically change their 3D shape. They uncoil or fold up when they absorb very specific wavelengths of light, like a blue photon.
Oh, interesting.
And third, you use the principles of photolithography, which is how we currently use lasers to project microscopic stencils to etch patterns onto silicon wafers.
So how do you actually merge a light-responsive plant protein with an indestructible prion?
You use genetic engineering to create a chimeric fusion protein. You literally splice the DNA code for the prion's cross-beta structure together with the DNA code for the light-responsive LOV2 domain.
Producing one single engineered protein molecule.
Yes. And the trick here is steric hindrance.
Steric hindrance. Sounds like a physical block.
It is. In its default state, in the dark, the light-responsive domain wraps around the prion domain, basically holding it hostage. It physically blocks the prion from interacting with anything else. So if you have a massive bioreactor filled with this engineered protein, as long as it is in the dark, it just sits there as a totally harmless inert liquid soup.
But the second you introduce light,
The second, a specific wavelength of light, say a precise blue laser beam, strikes that molecule, the photon transfers its energy. The LOV2 domain instantly alters its chemical conformation and uncoils.
it opens up the locked door.
Exactly. This sudden movement exposes the hidden prion domain. The prion is freed, and the instant it is exposed, it snaps into its rigid cross-beta structure, initiating the unbreakable hydrogen bonds and starting the self-assembly cascade.
But, and I think this is the key here, it only crystallizes exactly in the precise three-dimensional space where the photons intersected.
Exactly.
It is revolutionary. You aren't just turning a cellular process on and off. You are using light to write physical, solid architecture into a liquid medium with literal molecular precision.
It's a 3D printer where the ink is a living biological fluid and the print head is just a holographic projection of light.
That brings us right back to the factory vat from the intro. It completely redefines manufacturing. And Enfield provides incredibly vivid examples of how this will be applied. Let's return to the medical field for a moment.
Okay.
Imagine a patient with a deep, inoperable, solid tumor. A surgeon can't cut it out without destroying vital organs. Under this new paradigm, the physician injects a viral nanite carrying the genetic code for this optogenetic prion construct directly into the tumor.
The tumor cells are hijacked.
Right, and they begin manufacturing massive amounts of this liquid chimeric protein. But because the inside of the human body is completely dark, the protein just safely pools as a liquid inside the tumor.
And then the physician inserts a tiny fiber optic endoscope near the tumor and projects a highly precise 3D holographic laser pattern directly into the tissue.
Exactly. Wherever the laser intersects within that fluid, the protein instantly crystallizes into an indestructible amyloid scaffold. The doctor is literally using light to draw a physical, rigid cage inside the tumor in real time.
And if that amyloid is engineered with those piezoelectric properties we discussed earlier,
The doctor removes the laser, turns on an external ultrasound wand, and the newly formed scaffold generates massive voltage, releases reactive oxygen species, and utterly vaporizes the tumor from the inside out.
it's breathtaking. But what's even wilder is how this exact same technology translates to macro-scale manufacturing in agriculture. Enfield refers to this as the replicator paradigm.
Like Star Trek.
Very much so. And he uses the example of printing meat. We hear a lot about lab-grown cultured meat today, but right now it is mostly just an amorphous protein mush.
Yeah, it doesn't have the bite of a steak.
It lacks texture because real muscle tissue has incredibly complex 3D microarchitecture fascia, connective tissue, perfectly aligned muscle fibers.
Yeah.
You can't easily extrude that through a mechanical nozzle.
But with optogenetic lithography, you don't need a nozzle. You fill a massive transparent vat with a liquid bath of muscle cells, fibroblasts, and the optogenetic scaffolding proteins.
Then you simply project a high-resolution holographic light pattern of a perfect steak's cellular geometry directly into the vat.
Instantly, the structural scaffolding crystallizes along the light paths, providing the exact micromatrix for the muscle cells to align and bind to.
No animals are slaughtered. No immense tracts of land are cleared for grazing. No physical molds are used. You use light to define the geometry of food.
And think about how this connects back to the macro scale we started with. You use the massive duckweed and algae bioreactors we talked about in Section 1 to capture CO2 in agricultural runoff, generating millions of tons of raw, cheap protein feedstock.
You dump that feedstock into a massive synthesis vat.
And then use lasers to instantly grow carbon-negative aerospace panels, or frictionless engine components, or biocompatible heart valves. You eliminate the need to mine metal, you eliminate smelting, you eliminate toxic chemical processing. It drops the marginal cost of producing complex physical goods to near zero.
It is the ultimate manifestation of the biological loops. We are harvesting sunlight and waste and computing it directly into physical structure.
But again, my brain naturally goes to the potential failure modes. I need to understand the boundary limitations of this.
Naturally.
If I shine a laser into the vat to initiate the prion cascade, and the protein starts snapping into that rigid structure,
Right.
what stops the fiber from just continuing to grow right out of the illuminated zone and into the dark areas?
Right, the cascade.
Yeah, if the prion growth is an exponential cascade where one protein converts the next, wouldn't the crystallization just keep spreading until it consumes all the liquid in the entire vat? Your perfectly sculpted, microscopically precise steak would just keep crystallizing until it turned into a massive, solid boulder of protein.
That is known in the literature as the cascade amplification problem. And it is the primary engineering hurdle of this technology. If you can't stop the cascade, you have zero resolution.
So how do they stop it?
Enfield details several solutions currently being deployed to constrain the growth. The most basic approach is rigidly limiting the monomer concentration in the fluid, basically starving the reaction at the borders.
Okay, that makes sense.
A more advanced method is introducing competitive quenching agents into the vat. These are synthetic molecules designed to constantly hunt for and cap the growing ends of the amyloid fibers. The fiber can only continue growing if the intense light energy is actively overpowering the quenching agents in that specific spot.
But there is a more elegant, actively managed solution he mentions too, the Phobit 1 system. How does that work?
Phobit 1 is brilliant because it introduces a highly modular light-off switch. Instead of just relying on chemical competition, you engineer the system to respond to two different colors of light. The primary wavelength, say, blue light, causes the LOV2 domain to uncoil and initiates the rapid crystallization. But you introduce a secondary protein component that responds exclusively to red light.
And what does the red light do?
When hit with red light, this secondary protein instantly binds tightly to the end of the amyloid fiber, actively halting its growth.
So you aren't just projecting the image of what you want to build.
Precisely. Your holographic projection is dual wavelength. It projects the blue light to draw the interior of the object while simultaneously projecting a tight, microscopic containment border of red light completely around the edges.
That is amazing. The red light actively dissolves or caps any fiber attempting to grow beyond the defined boundaries. It gives you absolute, crisp, molecularly sharp spatial control over the biological cascade.
That is just phenomenal engineering.
It really is. So let's take a deep breath, step back, and look at the totality of what we've assembled here. We have a full, comprehensive technology stack. We have the viral nanites and CRISPR to precisely target and edit the code of life. We have the engineered prion amyloid scaffold acting as the ultimate tunable, indestructible building material.
We have optogenetic lithography, allowing us to print and shape that material with molecular precision using just light.
And we have the planetary bio-CCS and duckweed loops to power the entire ecosystem using atmospheric carbon and waste runoff.
It is exactly as Enfield titles it, a unified architecture. Biology is the physical hardware, light is the operating system and the programming language. And the application you can run on this stack is literally anything you can imagine.
From a completely healed underground aquifer to a cured genetic disease to a terahertz DNA supercomputer, we have the tools to completely author our physical reality.
But as we transition to section 5, the synthesis, Enfield forces us to confront a very heavy reality.
Yeah.
Having the power to redesign civilization and rewrite human biology from the ground up does not automatically mean we have the wisdom to wield it responsibly.
Right. And this is where the conversation pivots from the cool mechanics to the profound ethical stakes. Because if you are listening to this, the obvious question is, what does this actually mean for you?
What happens when programmable evolution becomes a tangible reality in our lifetimes?
Enfield issues some severe warnings in this final section. He argues that if this technological stack follows the historical trajectory of typical pharmaceutical development, where massive corporations hold monopolies and the therapies become exclusive luxury markets for the highest bidder, we are going to witness a terrifying divergence in the human species.
We will move far beyond simple economic inequality. We will enter an era of permanent biological stratification.
It's a dark thought.
Picture a reality where the ultra-wealthy can afford regular viral nanite infusions. They purchase the tardigrade radiation resistance package, they permanently optimize their oxygen efficiency, they edit out any genetic predispositions to cancer, and they install biological routines that dramatically slow cellular aging.
Their children are born inherently stronger, smarter, and more resilient.
Meanwhile, the global poor are completely locked out of this architecture. They are still suffering and dying from untreated malaria, basic heart disease, and unedited sickle cell anemia. We wouldn't just be divided by wealth anymore. We would be creating two entirely separate, diverging biological classes of humans living on the exact same planet.
It sounds like the ultimate dystopian sci-fi trope, but Enfield's entire point is that the underlying science for this isn't centuries away. It is arriving right now in FDA trials. The train is leaving the station.
So he demands that we establish incredibly strict global safeguards immediately.
What are the rules of the road for a species that suddenly discovers how to author its own biology?
He lays out a very strict ethical framework. First, he insists on an ironclad principle of treatment before enhancement. The entire biological platform must be dedicated to eliminating baseline human suffering before any elective augmentations are legally permitted. We must fully deploy the architecture to cure sickle cell, to halt Alzheimer's, to reverse cystic fibrosis. The clinical infrastructure and the delivery safety must be rigorously proven in the pursuit of healing before we ever open the door to enhancing healthy individuals.
Second, he argues passionately for a total global commons approach to the intellectual property.
Yes.
He states that the foundational tools of life, the CRISPR mechanisms, the viral delivery vectors, the functional translation libraries, cannot be locked behind corporate patents. You cannot allow a single biotech firm to essentially hold the copyright on human genetic augmentation.
Or the primary biological manufacturing platforms that will replace industrial agriculture. It has to be treated as a public good, like the GPS satellite network or the foundational protocols of the internet.
And third, which is arguably the most critical legal boundary he proposes, there must be a sharp, heavily enforced distinction between somatic interventions and germline interventions.
Let's clearly define the biological difference between those two for a second, because it's vital.
Okay, what is a somatic intervention?
A somatic intervention targets only the fully differentiated adult cells of the specific patient receiving the treatment. If you receive a viral nanite infusion that permanently optimizes your liver to reduce cholesterol or gives your muscle tissue enhanced oxygen capacity, that genetic edit is completely confined to your body.
So when you die, that specific genetic code dies with you.
Exactly. It is a closed loop. It is your choice, affecting only your physical form. A germline intervention, however, means editing the DNA of a human embryo or altering the sperm and egg cells.
Meaning the edits are heritable, they get passed on.
Exactly. Any change made to the germline will be permanently woven into the human genome. It will be passed down to that person's children and their grandchildren, persisting in the human gene pool forever.
So what's Enfield's stance on that?
Enfield argues forcefully that germline interventions must remain under a strict, impenetrable international moratorium. We simply do not possess the ecological or evolutionary wisdom to make permanent, irreversible biological decisions on behalf of countless future generations who cannot consent to the code we write for them.
That distinction makes perfect sense. Reversibility and humility are massive themes woven throughout his essays. It's why he strongly prefers transient mRNA delivery systems over permanent DNA integration when possible. The mRNA executes its function and degrades in a few days. You get the benefit of the biological app, but you don't permanently alter the hard drive unless absolutely necessary to cure a fatal disease.
All right.
But I want to push back on the fundamental underlying philosophical premise of Enfield's entire architecture here.
Go ahead.
If we start treating the Earth, our oceans, our crops, and our own bodies like programmable machines, if we view biology purely as code that we can optimize, patch, and rewrite to suit our needs, aren't we displaying the exact same arrogant hubris that got us into the climate and ecological crises in the first place?
It's a fair point.
Isn't replacing a mechanical factory with an engineered prion vat just doubling down on our desire to dominate the natural world? Aren't we just finding a more sophisticated way to play God?
That is the deepest, most persistent critique of synthetic biology, and it's a completely valid fear. If we simply swap out mechanical extraction for biological extraction without changing our mindset, we will just create new, unprecedented ecological disasters.
Right.
But Enfield counters this critique with his framework of planetary citizenship. He points out the reality of the Anthropocene. We are already altering the entire planet and our own biology on a massive scale. We're just doing it blindly, clumsily, and destructively through the linear fallacy. Planetary citizenship isn't about dominating nature, it's about paying our biological rent.
Paying our biological rent. I really like the phrasing of that.
It goes back to the concept of mass balance accounting applied to deep ecology. Right now, we extract million-year-old groundwater, we burn fossilized carbon, we dump synthetic nitrogen into our rivers, and we confidently label it economic growth.
But in reality, we are just liquidating the Earth's natural capital and pretending it is income. We have run up an absolutely massive biological debt.
Yes. Enfield's argument is that programmable biology, using vast duckweed networks to recycle nitrogen, engineering algae to pull carbon out of the sky, and utilizing precision genetics to heal ecosystems rather than poison them, is the only mathematical way we can actually start paying that debt back.
So we aren't playing God. We are finally accepting the responsibility of being conscious, active inhabitants of a deeply interconnected living system.
We are moving from acting as a destructive parasite to functioning as a symbiotic partner.
It is a profoundly beautiful, hopeful vision. To quickly recap the incredible journey we have been on today, we started at the planetary scale, looking at how the linear fallacy is draining the Ogalla aquifer, and how humble duckweed and photosynthetic algae can actually rebuild those broken loops to capture waste and generate clean power.
Then we zoomed into the microscopic level.
Right. Learning how to debug human diseases at the genetic source using viral nanites and CRISPR text editors. We explored how to strip the pathogenic nature out of prions to build indestructible, electrically conductive amyloid fibers that can wire up terahertz-speed DNA supercomputers.
And finally, we discovered how we can print those structural materials into any shape we desire, from medical scaffolds to lab-grown muscle tissue, using nothing but vats of liquid protein and holographic lasers.
It is a breathtaking synthesis. He takes incredibly disparate fields, hydrology, virology, quantum biology, optics, and proves that they are not separate sciences at all. They are all just different layers of the exact same programmable biological architecture.
It really changes how you see the world.
And so I want to leave you with one final lingering question to mull over as you go about your week. It's inspired by this deep dive, but it takes the philosophical implications just one step further. If everything Enfield predicts comes to pass, if our bodies, our food supply, and our physical environment all become fully programmable, if the code of life is truly just another software platform that we can patch, update, and rewrite at will, what happens to our fundamental concept of nature itself?
That's a huge question.
Right. If we successfully author our own biology, can we ever go back to just being human? Or are we forever burdened with the terrifying, beautiful responsibility of being our own creators? That medical X-ray machine we use to view our broken bones, we aren't just passively looking at the picture anymore, we are holding the pen that draws it.
We are holding